So what I did with that long, made it disappear. Here we go. What I did is I did a Google search on Mendelian genetics problems so that we could basically go through these things and you could see what the process is. Also, the process that I go through in order to solve these kinds of things. I'll make this bigger. I'll try to. Oh, there we go. So these are Mendelian genetics problems. This is simply the first website. I typed in Mendelian genetics problems. The first website is the one at the top here. I'm going to turn on the light so that the board is more, oops, not that light. So the board is more visible. Oh, not that light either. That light. And grab a piece of chalk. It turns out these are not colored chalks. I thought for sure that must be a pink colored chalk, and it is a pink colored chalk, but it's not. In fact, this is an example of a phenotype, right? The color of the chalk. But in any case, it's what something looks like, looks broadly defined. Okay, so the gene for hair color in rabbits has two alleles, big Q and little q. And big Q is dominant and codes for brown hair. So Q is the genotype, or at least an aspect of the genotype. Gives rise to brown. Why it's not a big B rather than a big Q, I don't know. And it is dominant to white hair. So little q is white hair. So since we have a dominant, we've been told it's a dominant recessive relationship, and actually, to back up, often white is the recessive because what white represents is a lack of color, and what a lack of color typically will represent is a lack of a protein that normally would be making the color. So this individual, if they have two of these, they're little q, would have at that specific Q locus two different alleles, different meaning that they're physically different, separate from one another, that both encode a protein that is probably not functional. So as a consequence, these guys are white in terms of their phenotype. This is their genotype. And that's their phenotype. Now, somebody who has a big Q allele possesses at least one allele that is capable of making a functional protein that is able to generate this color, the brown color. Now in this case, because it's a dominant recessive relationship, just having one of these proteins, functional proteins, is enough to give you the full-blown phenotype. And as a consequence, big Q and anything else gives you brown. So therefore, big Q gives you brown, and big Q, little q gives you brown. Now, if you look at this individual, you see a white rabbit. That is displaying the homozygous recessive phenotype. And since if you had a big Q in there, it would be brown. Therefore, this thing has to be a little q individual. So if you see the recessive phenotype, you know what the genotype of that individual is. So recessive phenotype for sure gives you homozygous recessive genotype. Now it doesn't give you it, it just means that you know if you have a recessive phenotype that that individual has to have the homozygous recessive genotype, which in this case is little q, little q. So phenotype. In reality, genotype is what gives rise to phenotype, not the other way around. But when you're doing Mendelian genetics, you're going from that to that. You're trying to infer what that must be from this. Now, these guys, the brown ones, since they can be either a big Q, or big Q, little q genotype, that means that when you see a brown individual, you don't know what its underlying genotype is. It could be either big Q, or big Q, little q. So today, we have probably knowledge of, assuming that the rabbits have been sequenced, I can't imagine they haven't been. We probably have knowledge of where the gene is located that encodes the color of their fur. And so today we could probably just go in and sequence that gene and see whether or not they have a little q or a big Q allele. Or we could use something called polymerase chain reaction and have the experiment set up in such a way where you don't get a product, a DNA product, an amplicon, if it has the little q allele, or you don't get a product if it has the big Q allele. But you do get one if it has a big or whatever. They've got it set up so you can distinguish between the two easily if you know what the actual sequence is to begin with. But back in the day, the only way you could figure this out was by doing crosses. So with all of that, we have write out the possible genotypes and phenotypes. So we've written out the possible genotypes and phenotypes. 1, 2, 3 are the genotypes, and brown and white are the phenotypes. So that's it for the first one. Okay, so notice that, I mean, it's easy, hopefully, but notice that if you're given phenotype, you want to figure out what genotype is. If you're given genotype, you want to figure out what phenotype is. Now, that really didn't make you do that, but maybe the next one will. So using, in fact, let's get rid of the answer here. I think we can. Not make it bigger. Okay. Using the above example, fill in the Punnett square of offspring genotypes if one parent is heterozygous and the other parent and the other is white-haired. So here, that's a description of genotype, okay? And that's a description of phenotype. But we know if they have the homozygous recessive phenotype, we know what the genotype is. But they're telling us that the other individual, in fact, they're not even telling us what this individual's phenotype is, are they? But we know that if you don't have 2 recessive alleles, then it's going to be brown. Heterozygous means you don't have 2 recessive alleles, and in fact means this here. And that's the white one. So that's one parent, that's another parent. So now they want us to make a Punnett square. So we're going to cross big Q, little q individual that is going to generate a gamete that is, half their gametes will be big Q and half their gametes will be little q. I'm going to cross that with a individual that can only make little q gametes. And the Punnett square looks like this, but in fact we don't need to even make it into a square because these are going to have the same genotypes that are filled in, and those are. This big Q, little q, meiosis. And this little q, that's the parent. And that too is the product of meiosis. And so here what we're doing is we're filling in the products of fertilization. So we've got the gametes. And now we're going to get those gametes together. And then if you have a large enough number of gametes and you're mixing them all together, you'll end up with big Q, little q, little q, little q. And then this is just redundant on the bottom because this individual can only make little q carrying gametes. And so this means that statistically half of the gametes, half of the products of fertilization, therefore half the offspring, are going to be heterozygotes, and half of them are going to be homozygotes. As it turns out, this is the same genotype as one of the parents, and this is the same genotype as one of the parents. But when we did this cross, if we didn't come up with 50-50 in terms of this would be brown, and this would be white. If we didn't come up with 50-50 brown and white, instead we came up with 100% brown, then we would know that this wasn't a cross between a heterozygote and a homozygote. It would be a cross between a homozygous recessive and a homozygous dominant. Because this individual, therefore, if it were the case where they were homozygous dominant, would only be able to make big Q gametes. And therefore, the result would be just that, which would be 100% instead of 50% big brown. So let's see, what does it say? The pair of rabbits had a litter of 24 babies. Write out the expected number of each genotype and phenotype in the table below. So this is an example where, because it's going to be 50-50 in terms of phenotype, and because of the way we did what the cross consists of, the phenotype maps directly onto genotype numerically and in both of those cases. But nonetheless, we're not always going to have the phenotypic ratio and the genotypic ratio being the same, but in this case they are. So what's our genotypic ratio in this case?
1 to 2. Technically 12. I mean, there'd be 12 of each.
Now you're getting ahead. What's the ratio?
Is it 1 to 1?
It's 1 to 1. Yes, that's the phenotypic ratio. What's the genotypic ratio?
Would that be why the 12 at 1 to 1?
Yeah. Yes. They're both 1 to 1. 50-50. I mean, that's what a 1 to 1 ratio means. Oh, that's a terrible 5. 50-50 is the same thing as 1 to 1. 50%, 50%. Now I know you're saying 1/2 of them are brown and 1/2 of them, that's where the 1 to 2 came from. But it's actually a 1 to 1 ratio. So, since you've already answered it, what's the answer to the question?
The 12 are going to be the I don't know, white, and then 12 will be brown, right?
Okay, that's one of the answers. They're asking for another answer here, though.
There'd be 12 genotype and 12 phenotype. I don't know. Let me read the question. That might help. Right, so there'd be 12 white and then 12 brown.
Okay, that's half the question.
I don't know.
It says the expected number of each genotype and phenotype. So you answered the phenotype part of the question. What's the genotype part of the question?
I don't know the answer.
Half of them would be big Q, little q, and half of them would be little q, little q. So all of this, except for that right there and that right there, is genotype. And then genotype is going to give rise in some perhaps simple way or perhaps in some complicated way to phenotype. And so in this case, genotype, it could be big Q, big Q, big Q, little q, little q, little q. And phenotype can only be brown or white. And because we decided that there's going to be no big Q's involved, then if our phenotypic ratio is 1 to 1, then our genotypic ratio is 1 to 1. And it's important to point out what the actual genotypes are when you're answering that. That all make sense?
Yep.
Okay. There we go, look at that. Let's not go to the next one yet, that we don't see what the answer is. So the gene for plant height in sunflowers has 2 codominant alleles. Okay, so now we already saw a dominant recessive relationship. And so that means that the little genotype is going to be whatever the recessive phenotype is. And as long as there's one big allele in there, you're going to have the dominant relationship. This creates problems because you never can know whether or not they're the dominant homozygote or a heterozygote, unless you do a crosser in some other way figure that out. Codominance means that you get a phenotype for the heterozygote that's different from the phenotype for the homozygote dominant. So it might be that this is red and the recessive is white and you've got pink as the intermediate. But here it's probably going to say that if you've got the dominant allele, you're big. If you've got nothing but, 2 dominant alleles, you're big. Nothing but the recessive alleles, you're small. And if you're a heterozygote, somewhere in between. So it says T sub 1 for tall plants and T sub 2 for short plants. If the tall plant is crossed with a short plant, fill in the table below. So I suspect that what they're saying here is that the tall plants are not intermediate plants. So therefore, they must have 2 dominant alleles, so 2 T1s, and the short plants are not heterozygote, so they must have 2 T2s. So this cross therefore, we have a T1 individual, and we're crossing them with a T2 individual, or at least that's how I'm reading the question. I might be reading the question wrong, but that's how I'm reading it. So we'll put that individual here. So that individual can only give rise to T sub 2 gametes. And that individual can only give rise to T sub 1 gametes. And so we can make the Punnett square. And therefore, when fertilization occurs, we get a combination of T1 and T2, which is the heterozygote. And it would be the same for all of those boxes. So what would be the ratio here of the phenotypes, of the genotypes, rather? That's a tough question to answer, but it'd basically be 1 to 0. I know we don't like putting zeros in fractions, but that's essentially what it would be. You would get 100% of them would be that. And therefore, since you only have one genotype, 100% of the offspring would also be that phenotype, assuming we don't have complicated genetics here. So let's see how they answered this. Yeah, so that's what we. That's the answer that I put up there. 100% are that genotype, and it does say there that means that their phenotype is medium height, and so that goes back to this idea of codominance, that these guys are tall, these guys are short, and these guys are somewhere in between. When you have something other than a complete dominant recessive relationship, then when you see the phenotype, you know what the genotype is. And that makes, of course, doing all of this much easier. It's really when you have the dominant recessive relationships that it becomes difficult. Okay, so take any 2 of the seedlings from part 2A and cross them, fill in the results below. So now we've got those guys, only those guys, T1, T2 crossing with T1, T2. So these guys are heterozygotes. It's a monohybrid cross. It's hybrid because it's the heterozygotes, and it's mono because it's only one locus that we're looking at, the T locus. So you cross these guys, so they make their gametes, meiosis. Gametes are either T1 or T2. It's the same for those guys there. In this case, you really do need a Punnett square. Fertilization, in this case, would be a T1, T1 individual. In this case, it would be a T1, T2 individual. In this case, it would be a T1, T2 individual. And in this case, it would be a T2, T2 individual. So we've got really a dominant homozygote there. We've got a recessive homozygote there. And then we've got two, and I'll do it a different shade. We've got two heterozygotes. So what would be the ratio here? You don't answer.
1 to 2 to 1.
Yes, 1 to 2 to 1. So whenever you have a monohybrid cross, and just looking at genotypic ratios, you end up with a 1 to 2 to 1 ratio. Dominant homozygote, heterozygote, recessive homozygote. Now, in this case, we're talking about codominance. So what is the phenotypic ratio? You can answer this time, but you don't have to. So the genotypic ratio is 1 to 2 to 1. There's a difference in terms of phenotype between all of the possible genotypes. Remember, they're tall, they're short, and they're somewhere in between. Phenotype is what the organism looks like or is like. It's measuring something other than the genotype of the organism. So given that there is a phenotype possibility of being tall, of being medium height, and being short, what is the phenotypic ratio that we're seeing here? Anybody can answer.
So one short, one tall, and then two in between.
Yeah, it's a 1 to 2 to 1 ratio. So in this case, when we have codominance, the genotypic ratio from this monohybrid cross is a 1 to 2 to 1 ratio. The phenotypic ratio is also a 1 to 2 to 1 ratio because we can distinguish all of the different genotypes phenotypically. If this were instead a complete dominance relationship where T1 was dominant to T2, what would be the genotypic ratio? It would be the same thing. We didn't change anything. It's the exact same cross. When we're talking about genotype, we're talking about the alleles. So it's a 1 to 2 to 1 ratio. But what's the phenotypic ratio?
Same thing.
No. Because we went from a co-dominant relationship where all the phenotypes, all the genotypes had a different phenotype, to a complete dominant relationship where the recessive homozygote has a specific phenotype, but the dominant phenotype can be more than one genotype. So if T1 is dominant to T2, what's the ratio of tall to short, or I guess brown to white? We'll go back to the rabbit example.
Like 3 to 1.
It would be 3 to 1. So anytime you are seeing these 3 to 1 ratios, your first thought should be, oh, it was a monohybrid cross and it's a complete dominant relationship. That's in terms of phenotype. anytime you are seeing a 1 to 2 to 1 ratio, you should be saying to yourself, oh, it's codominance that we're looking at, and the cross was a monohybrid cross, a cross between two heterozygotes. That's the absolute basics of what we're doing. Everything else is just building on that basic idea. If you have a cross between two homozygotes and they are both recessive, then you get nothing but recessives. If you have a cross between two homozygotes and they're both dominant, you get nothing but dominants. If you get a cross between a homozygote and a heterozygote, that was our first example, and so on and so forth. So you want to know, and you might not know to begin with, you want to know what the genetics are, you want to know what the genotypes are, and therefore you can go through the meiotic step and generate one of these Punnett squares and you get what your expected ratios are. Alternatively, you could be looking at the ratios in the offspring and work your way backwards to trying to figure out what the genotypes of the parents must have been and what the genetic relationship between the alleles must have been. So that's that one. And so let's make sure we got that right. So that's their cross, that's the monohybrid cross, and that's our 1 to 2 to 1 ratio. In fact, here's our 1 to 2 to 1 ratio. Yeah, we're not going to do sex chromosomes because we're not going to go to someplace. But this one's actually interesting, the color blindness part 1. Okay, so that's not looking at sex chromosomes. It's looking at autosomally encoded traits. So they're not on an X or Y. For some reason, I think I could figure out why. I was looking at papers, or a paper, on evolution of sex ratios. And in, well, I'll say turtles, but it's more than just turtles. Turtles, whether the offspring comes out as male or comes out as female, is dependent on the temperature that the eggs are incubated at. And apparently with global warming, it means that that temperature might change and you might end up with a skewed sex ratio, which of course could be problematic. Sex ratios want to be, in the sense that they will tend towards, a 1 to 1 ratio. And that's because if you're male and there's lots of females, you're better off being, if there are more females than males, then you're better off being male because it'll be easier to find a female. If there are more males than females, then you're better off being female because it'll be easier to find a male. And so that ends up balancing that logic so that you end up with a 1 to 1 ratio. In humans, males are the weaker sex. We tend to die off more easily, actually, for reasons that are relevant to this next question. And so we actually produce at birth more males than females. And the idea is at the point of sexual maturity, the ratio will have evened itself out at 1 to 1 because the excess of males will have died off, simply because they're genetically weaker than the females. They have a higher likelihood of dying. In any case, 1 to 1 ratios. But color blindness is a recessive trait. So recessive, that means that we're going to have A. Yeah, we do have A. So if it's recessive, that means big A equals not colorblind, and that's an A. Little a means colorblind. I'll just say CB. But this is not an autosomal trait. This is a trait that is carried on a sex chromosome this X chromosome, which is one of the sex chromosomes, is much larger than the Y chromosome. So most of these traits that are sex-linked are associated with the X chromosome simply because the X chromosome has so many more genes than the Y chromosome. Also, the females don't have Y chromosomes. So if you have something on the Y chromosome, it's only going to be seen in the males. So the focus on traits that are inherited on sex chromosomes is on the X chromosome. So this is, of course, is very basic. So XX is female, and XY is male. And of course, the mother can only give an X to her offspring, but the father can give either an X or a Y. I'm making sure that my X's and Y's look different. And therefore, it's the sperm that determines the gender of the offspring. So that's a different approach to determining gender than temperature, and there are other approaches that different organisms take. So the question is, color blindness is a recessive trait caused by an error on the X chromosome. The big A is normal vision and the little a is colorblind. If the mom is normal, okay, so this is a complete dominant recessive relationship. That means that to be not, in terms of a diploid genotype, that individual can either be big A, or big A, little a, because it's a complete dominant relationship. Now it's on the X chromosome, so we have to put in the X's here. But when this says that the mom is normal, meaning not colorblind, you stop the question right there, okay? And just go to there. She could either be big A, or big A, little a. However, going further, it says that she's not a carrier. So if she's not a carrier, the meaning of carrier is that she's a heterozygote, which doesn't display the actual phenotype associated with the recessive allele. So if she's not a carrier, what they're doing is telling us that she's not a heterozygote. And dad is colorblind. Why does dad have to be colorblind? Because if dad wasn't colorblind, this would be a truly boring, meaningless question. But they have to make dad colorblind. So that means that mom is just going to be able to supply the X sub A. Yeah, sounded good. But dad is going to be able to supply an X little a and a Y, which has nothing to do with this color blindness thing. So that means that all of the girl offspring are going to be carriers, and all of the boy offspring, and they can't be carriers because they can't be heterozygotes for these genes, are going to be normal. Does that make sense?
So you're only ever going to have a colorblind female? Like that? If the mother is not a carrier and the dad is a carrier.
No, you said it slightly backwards. You're only going to have a colorblind female if the mother is a carrier. In fact, that's kind of the meaning of carrier, right? And the dad has the trait, is colorblind. Which means that, well, this gets it deeper than into Mendelian genetics. It gets to Hardy-Weinberg stuff. But what that effectively means is that the likelihood that the female is going to be colorblind is much lower than the likelihood that a male is going to be colorblind. And that in turn is going to be dependent upon the frequency of the color blindness allele in the population. So if the frequency of the color blindness allele in the population were 0.1, 10%, then you'd expect 10% of the males to be colorblind. But you would actually expect 1% of the females to be colorblind, which is why, and the frequency is much lower than that in the population, which is why when you see an individual who is colorblind, they're likely to be male and unlikely to be female. It's just all statistics. That's Hardy-Weinberg stuff. So that's population genetics, which is studied by evolutionary biologists. In any case, actually, let me pause. I mean, are we ready to move to the next step? Okay, so that should look like what I just put up here, or I should say, that should look like what I just put up here, and it does. And then they are talking about what the phenotypes they expect here. So she would be a normal, but she would be a carrier, and he would be normal, which is what we concluded over here. They're just not noting that she would be a carrier. So let's see. None of the there it is. None of the children be colorblind, but the girls are carriers and can pass it down to half of their children. Now, when they say pass it down to half of their children, that does not mean that she is going to pass down that allele to half of the children who will end up being colorblind. It means that she's going to pass down the allele to half the children. So if she marries a normal male, half of her children are going to be carriers. I'm trying to make sure that that statement is correct. Half of her girls are going to be carriers, and half of her boys are going to be colorblind. In fact, let's do that. So so we've now generated big A, gotta make the X big, little a. And here we're assuming that she's mating with a normal male. So that's a male, big A, he's normal. That's the female. So in this cross, big A, big A, big A, little a again, big A, and then that has to be changed to little a. So that means that 50% of her girls are going to be carriers, and 50% of her boys are actually going to be colorblind. And so that means that 50% of her offspring will have had the allele passed on down to them. So that's probably why they don't say anything about phenotype here, because it's a little bit more complicated, because there would be both carriers and individuals that are displaying the trait. So let's do the same problem again, but this time with a carrier mom and a normal dad, which we just did. This is going back to what I've been trying to emphasize, that the way to solve these things, you just want to play. You want to say, okay, you got a phenotype, what's the genotype? Or you got a genotype, what's the phenotype? Let's figure it out. What could this mean? Where can we go with this? So, I mean, I'm just playing up here. It's obvious that we wanted to figure this out, right? And that's the very next question. So I already did that. So that is, or should be, the same as that. And the conclusion, so that's, again, just making sure I emphasize that. Genotype and phenotype. So half of the females are normal. All the females are normal, but half of them are carriers. half the males are normal and half of them are colorblind. By the way, I've been struggling with this colorblind thing because I've got these graphs that I'm making that have, what was it? I think I showed this to you last week, have 27 different curves on them, and I want to distinguish them in some way, but at the same time not have this red versus green thing going on, because if you do that, then people who are colorblind aren't going to be able to tell what's going on. But even so, I still threw in a green curve that is kind of separate from the rest because I ran out of colors. So this is an ongoing problem. I mean, it's not a problem. It's something that one should always be aware of, and it's not always easy to address. Oh yeah! Ooh! Oh! 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 So this is what we just did up here. So this is well, I guess it's not entirely. They're not actually looking for it quantitatively, but there are more males in the population that are colorblind because half of the males in a cross like this are going to carry the colorblind allele, and all of them are going to be colorblind. Whereas half of the females in a cross like this are going to carry the allele, and none of them are going to be colorblind. And then we can talk about what the actual frequencies must be. Because colorblindness is carried in the X chromosome, and males have the X, they only need one copy of the defective X in order to be colorblind. Females have 2 copies. So we need 2 copies of the defective X in order to be colorblind. There are only 2 genotypes for males, but there are 3 genotypes for females. The dominant homozygote, the heterozygote, and the recessive homozygote. So males can't be carriers, they either have it or they don't. That's probably a more detailed answer than he would ever, or she, whoever put these together would ever get from a student, but that's the answer. Just going through this one to make sure that this isn't too hard. Oh, okay, we can do this. So in guinea pigs, 2 different genes affect the coat. One gene codes for coat color, saved by the bell.